Okay, I am going to go through some notes on cell signaling and signal transduction pathways with you. So if you struggled with your POGL or wanted more information on how cells communicate with one another, this is the spot for you. Um, we're going to just kind of start off with what a signal transduction pathway is and some of the basics of what you will see when cells are communicating with each other. So, signal transduction pathways are going to be a series of steps that lead to some sort of specific cellular response. In order for any signal transduction pathway to begin, there has to be this first step of reception. There has to be some sort of signal that is received on a cell's surface in order to begin this communication pathway. And those signals are called ligands, which hopefully you remember that from your POGL, but it's going to be a small little um, signaling molecule that will go through and attach to a protein that will start this pathway. So step one is reception. Step two is going to be the transduction, or the series of steps, kind of intracellular, to pass information along. And then once that information gets to its target area, that will initiate some sort of cellular response. And there are many different types of responses we can see out there, which I'll get into. So here's kind of the drawing visual for you. Hopefully this, again, is a reminder of that. A ligand is going to be some sort of binding molecule, and there's different types out there. They're going to have different shapes to them depending on what sort of cellular response they are looking for. Those ligands will fit lock and key with the receptor molecules or the receptor proteins that are on the surface of the cell's membrane. And then again, those receptor proteins are going to have a specific shape that are going to fit with that specific ligand. So there are many different types of ligands and receptors out there, and based on those shapes, and that will signal specific responses within the cell. Right. So this is step one, which it says signal perception, which is the same thing as reception. Hence the receptor molecules, right? So that's step one. Then down here, we can see there's kind of a multi-step pathway here, right? So a signal will be passed along to a certain protein. That protein will pass it along to another or multiples after that. So this is called transduction, when there is a multi-step pathway within the cell passing along some sort of signal. Okay, that's transduction. And then the third step is the response. What is it that the cell is being asked to do? And that could be several things. It could be, like we just talked about with mitosis. It might be encouraging the cell to reproduce or go through cell division. It might be asking the cell, and a really common one is, let's turn on a specific gene or turn off a specific gene for transcription, or the creation of certain genes. Oops, sorry. So that's another one. It could be signaling apoptosis and saying, hey, there's something wrong with the cell. We need to shut it down. Lysosomes come on through and start dissolving this cell. So there's lots of different responses that we can have triggered from this three-step process, but it all depends on what type of ligand is binding to a specific receptor in the beginning. So a common lab that we usually do is mating different versions of yeast with each other, or putting them near each other and have them signal with one another to show that they're communicating and show the new cells that are created from it. So there are two different types of yeast cells out there. Unfortunately, we can't do this one because it takes multiple days, and that would mean that we're intermingling supplies with each other. So that's a no-no, but there are two versions of our yeast. So there's yeast A and there's alpha yeast over here. And they're each going to have their own mating factors represented by these circles and squares for their ligands or their signaling molecules. So if they are near each other, there can be an exchange of these mating factors or these ligands and signaling molecules, because on each of these yeasts, they're going to have different receptor molecules, so they can pass them back and forth with each other so that they are received properly. Now, there might be not just only square receptors over here, they might have, receptors that are also circular, and vice versa over here, even though my image right now only shows one of each. So technically, this A yeast molecule can send a signal to itself for those square receptors, and it can go through and reproduce with only a yeast. Alpha yeast can do the same thing, but if they're nearby each other, they can do this exchanging of mating factors, which is going to cause a slight shift in their configuration. So we can see that these mating factors have been exchanged with one another. And since they're near each other, they're going to signal and say, hey, we can kind of combine and fuse together here to create a new combination cell, which is what we see at the end. So this is a process of the ligands and reception, which is step one. There's going to be communication and this transduction pathway that will happen, which is causing this right here, this change, and a response is the new cell being made. So these signal transduction pathways and cell signaling are signs of evolution because of all the links between our different organisms. So all organisms will have these ligands, or they'll have signaling molecules, and they'll have receptors. And there are a whole variety of them, but the fact that each cell has this ability is going to tell us that there's some sort of link between them. There's, a common ancestor between our cells and their ability to have this communication going on. So again, there's another one here talking about bacteria that can detect population density that might lead to certain growth or reproduction, that sort of thing, or perhaps even like a specific special trait to them, like bioluminescence, that they might give off depending on how closely packed together they are with one another. So anyway, there's a link because all cells can communicate in some fashion, whether it's to themselves or to other neighboring cells. So here's another example of that, right? So we've got an initial rod-shaped cells. We can see that there's an accumulation or aggregation in process here. This leads to a spore forming and new fruiting bodies, or another version of asexual kind of reproduction that's happening here. So because there's communication happening, this can happen again on a local level or a neighboring level between cells, and it can happen at a long distance level as well. So remember our organelles, our cell organelles that we talked about. So animal cells can have their local or neighboring cell communication happen through gap junctions. Our plant cells have specific communication pieces called the plasmodesmata, right? So those are going to be very similar to each other, but one in plant and one in animal. We can have, short distance messenger molecules, so they may not be directly in contact with each other, like the gap junctions and the plasmodesmata. They might have just a little bit of space between each other. And so there are short distance messenger molecules, and then there are also long distance molecules, which are most commonly associated with hormones, where it's going to go within an entire organism, but it might travel from, one organ to the next organ system. It might go all the way throughout an entire organism. So There are local and long distance signaling. Here are the examples of our gap junctions. Again, cells that are right next to each other, neighbors, even touching each other. So here are animal cells with their gap junctions that they can communicate their growth factors or mating factors with each other. Or we could have the same thing with the plasmodesmata for our plant cells. They're literally touching each other and are in contact and are communicating with each other. This is a nearby one with the cell-to-cell recognition happening, but they may not be, actually linked together. They might have a little bit of space. So this is our ligand that can link up and then trigger a transduction and response within a neighboring cell that way. Again, another look at some local signaling. So there are different terms that we see with that. So if it's happening to the cell itself, one cell all by itself, that would be called autocrine. So happening with one cell, which I don't have a picture of on here, but paracrine is going to be a little bit more specific for distance. This is interesting. What did I just say? Paracrine. Oh, sorry, not paracrine is for neighboring cells, right? They aren't directly in contact with each other. So this neurotransmitter, if you can think about nerve cells as they interact with each other. So they have dendrites. Any of you in anatomy physiology might be fully aware of what I'm talking about, but nerve cells are going to have kind of their body to them and then a tail that's attached to them, or the axons. And then little dendrites that can send communication to a neighboring cell that has their cell body over here waiting for a response. Anyway, they're next to each other, but they are not directly in contact with each other, which makes it that paracrine signaling. And they can trigger some responses to those neighboring cells or pass the signal along all the way throughout a certain area. So, right, you touch a hot stove, it may take a second for it to kick in, but that's because these neurotransmitters are passing the signal along all the way through your nerves there and eventually gets back to the brain. And then that's going to trigger the response. Hey, get your hand off that hot stove. And then you pull away. And that's all through this local signaling that's happening. So that was paracrine. Endocrine is going to be our long distance signaling. That's going to be associated to hormones. And so we know, think like pancreas and insulin and blood sugar levels. This would be another example of that, where the pancreas, can release insulin, and that can travel to different parts of the body to balance it out. It could go to the liver, it could go to, further cells, but it's going to travel throughout the bloodstream and take a longer route. So these signaling molecules are the longest lasting of them all for our different versions because they have to travel the furthest, so they will survive the longest. But think endocrine and hormones, and those are linked together. So like I referenced, there's going to be the 3-step process for our cell communication. Step 1 is the reception with the ligand or our signaling molecule attaching to the receptor, specific for their shapes and their functions. So they're going to find their appropriate receptor molecule. Lock and load, and that's reception. After reception occurs, sorry, this is exactly what I was just saying, but I forgot that I gave you some notes on it. We have our 3 different types of receptors that are out there. So there's G protein coupled receptors, which are going to be associated to a specific type of energy that will link up with it. Instead of ATP, it's GDP, which are very similar. There are receptor tyrosine kinases, which are going to be probably the most common ones that you come across. Anything with the kinase as we talked about with the CDKs and cyclin in mitosis, right? Those CDKs are kinases. Those are specific receptor tyrosine kinases. Anyway, that's where phosphates have to be added in order to trigger that transduction and response. And then there are also ion channel receptors, which are going to be associated to allowing charged particles to pass through kind of a gated ion channel. So here's an example of some receptor tyrosine kinases, and this is way more than we really need, but it gives you the visual. I tried to copy over one of my original slides from PowerPoint to our Google Slides here, so it didn't quite work out. But again, here we're outside of the cell. Here's our signaling molecules or our ligands, these circles, right? We can see that there are binding sites or active sites on our receptor molecules, and these ones are specifically receptor tyrosine kinases. We know they're tyrosines because they're going to be labeled with the TYR next to each other for the tyrosine portion. Right, so they're hanging out there waiting for that ligand to link and receive each other. So once those signaling molecules hit their receptors, then these two proteins are going to come together and they form a dimer, or they get linked together. And that will happen on a couple different levels. And again, sorry, this is slightly shifted around, but we can see the addition of energy here, or ATP. Once that dimer forms, ATP comes and links up, and it's going to drop off some phosphates to these proteins or these kinase regions. Once those proteins are dropped off to this receptor tyrosine kinase, that's going to fully activate those proteins and then start triggering a response. So we can see that, hey, there's some relay proteins that will come and link up and start saying, hey, let's do this, which might be, reproduction. So that's kind of the breakdown of our receptor tyrosine kinases. But again, this is step 1, reception. Step 2 is going to be the transduction, or kind of the series of steps to get to a response. So we can see here's one step of the dimer being formed. Here's the next step of ATP dropping off phosphate. And then after those transduction steps, we have our third part, which is the response of the cell. So again, here's another look at it with a ligand-gated ion channel. Here's our ligand over here with the specific shape. The gate is closed because it's not linked up. Here are the ions that we're trying to send into the cell. It'll go on through and link, open up the ion channel, and the ions will come through and create a response. So once that ligand binds to the protein, then the ions can travel through the ligand-gated ion channel receptor. Once those ions enter into the cell, they will trigger a specific cellular response. And then once that ligand detaches over here, then the gate closes up again and the ions cannot travel through into the cell. So again, step 2 is our transduction, which I was showing you on some previous slides there. But the definition for that is it's going to be a cascade of interactions, a cascade of signals that are passed along to eventually their final target molecule. Oftentimes, this relay of signals from protein to protein is going to cause a change in shape for that protein so that it knows to activate and then send the signal on to the next step of transduction. So it's kind of like this multi-step pathway. That pathway or that transduction usually is going to amplify that signal to ensure that it's reaching the cell or that target molecule and ensure that response is passed along. And the more molecules involved in the transduction pathway just means that there can be even more regulation. So if molecule A over here is sending the signal to molecule B, right? And then there's C and D, and then eventually the response is over here, right? So we have our whole pathway. You can regulate and maybe have something that blocks that signal here so that it doesn't go on to C and D, right? Or you could have something that binds to amplify and say, hey, C, let's get the next signal on a little bit more. So there are many ways to kind of regulate transduction. Again, here's that step 2. We talked about reception happening. Here's transduction. So that relay of signal molecules, again, it could cause changes to signal, hey, passing it on to the next one in the pathway. This represents kind of that area where you could attach another molecule to block the signal or amplify it. And then typically there's, more molecules than this happening during the transduction portion. Our last step is response, right? So once these are relaying their information, they're going to pass it on, and this should really all be shifted over. But again, my pictures didn't transfer the way they should, but this square over here is going to represent the cellular response that it's asking for. So what does response mean? These are typically the most common examples of a cellular response that you're going to see. Genes turn on and off, or transcription will turn on or off based off of that response. It might ask it to make some specific proteins or specific enzymes within the cell. It might change the overall shape of whatever protein you're trying to get altered or modified. So those are really common. Again, reproduction or cell division is another one. So all sorts of responses. Here is another look at that again. It could be growth factors, like we talked about in mitosis. So those could link to the receptor molecule here. There could be lots of phosphorylation happening or dropping off of phosphates for our tyrosine kinases. Okay, so this would be transduction. And then there could be some sort of response down here, which is an example of transcription or turning on and activating a certain gene.